welcome back to ZOO3649 Evolutionary Genetics. We're at lecture 27, the second lecture in tree building, methods for tree building. In the lecture 26, we discussed parsimony, which is the simplest way to make a tree. Then we looked at distance, and now we're looking at a big one, and that is called maximum likelihood. So big that we've given a whole lecture just to maximum likelihood. So I keep telling you that third year is very different to second year, and I'm pretty sure that all of you have figured out why, because things get very complicated very quickly. And this lecture and the following lectures are absolutely no exception. If you were lost in the previous lectures, well, you have no chance of understanding what's going on today. Okay, so if you're lost, I suggest go back to lecture 1 and start again, because this is going to be examined, okay, and it's going to determine whether you pass or fail this module. Right, so let's look at maximum likelihood. Now, the first two were non-model based, okay, distance and parsimony were not based on any underlying model. We've been through models before. You know that there's the Hardy-Weinberg model for determining whether there's evolution. There's the Wright-Fisher model for determining whether evolution is caused by genetic drift. And then there's the neutral model for determining whether evolution is caused by selection. So now we have another model, okay? And this is a model for how DNA bases change, okay? And we're going to get in, and they're called substitution models. So for maximum likelihood and Bayesian, remember parsimony and distance could be used on many different kinds of characters. Likelihood and Bayesian are only for DNA sequences, okay? And DNA alignments where you have a homologous position that you know is homologous, because the position, the trait must be homologous to make a tree. So you've got a homologous position, and there you have the character states changing from A's to C's to G's. And the models we use are the rules for the change of those states. And we'll get into them later on in the lecture. Okay, so that's why we call these model-based inferences. All right, so what is the maximum likelihood? Maximum likelihood is actually a clever way. It's likelihood for what we're concerned at third year level. Likelihood is the same as saying probability. Okay, if I say, what is the likelihood that tomorrow will rain? That means I'm saying, what is the probability that it will rain tomorrow? Or what is the chance that it will rain tomorrow? You can say, 50-50, prof. Yeah, you're probably right. Maybe it will be sunny, maybe it will be rainy. Okay, but that's the probability. And maximum and the likelihood is the same as saying probability. But when we deal with models of evolution, and we are dealing with lots of different potential answers to this question. For example, the question, will it be raining tomorrow, is only two choices. Yes, raining, no, sunny, right? Whereas when we're asking, for example, we want to build a tree, we are saying, well, what are the different possibilities of having a tree? Well, there are many different possibilities, right? You can rearrange the individuals in the tree in many different ways. So, and each one of them has a probability, a likelihood. So what do we want to know? We want to find the tree that has the maximum likelihood, because that is the correct tree, okay? So we need to figure out the probability of something, okay, after the thing has happened, and maximum likelihood allows us to do that. What do we need to calculate the maximum likelihood? You need the thing that has happened, okay? So in other words, we are trying to reconstruct a tree. So the tree has happened. We haven't reconstructed it yet, but it has happened. Evolution made it, right? And the evolution has happened. So the tree process of the tree has already happened. All we want to do is try to reconstruct it. Okay? So the thing that has happened is the tree. Okay? We need the result of the tree. What did evolution create while it was happening. While that tree was happening, while the evolution was happening in that tree, what was created? DNA sequences were created, right? Genomes were created. And that is what we call the observed data. Our DNA sequences are the observed data, okay? The observed data is the result of the evolution happening. Because we would not have the observed data if the evolution didn't happen, okay? The thing that has happened is the evolution, so the tree, and the observed data are the results of that evolution. And then we need some rules to tell you how things happen. How these things that happen, the thing that has happened, the result of the thing that has happened, and the rules to tell you how things happen in general. Okay? And those the rules that govern how things happen is what we call the model. All right? And you only need those three things. And we can calculate the likelihood of the observed data taking into account the tree and the model. All right. So the likelihood of the observed data, and as I said, based on the observed data, in other words, the DNA sequences, many different trees have a likelihood, right? All trees have a likelihood, to be honest, right? But the likelihood can be different. Some have a low likelihood. Some have a high likelihood, right? So the ones that are grouping together, for example, dolphins, ichthyosaurs, and fish, because they look the same, those trees will have a low likelihood. So we won't consider that is the right tree, because what are we looking for? We are looking for the tree with the maximum likelihood. The tree with the highest probability of being the correct tree. That is what the maximum likelihood is all about. We want all of the trees have a probability. All of the trees have a likelihood. Or there could be millions of different trees. But there's only one with the highest. And that's the one we are looking for. Right? So the likelihood of the observed data is not a single number. It is a function. It's a curve. It's like if you have an x and a y, it's a curve with a function. And the top part of that function, the highest point of that function, is called the maximum likelihood. It's the highest point of the likelihood function. Okay? Because every possibility has a likelihood. Okay? Every tree has its own likelihood. We want to find the tree sitting at the peak, like that maximum, where the likelihood function goes to the very peak. It can go down again. We don't care. But we want the tree that is at the very peak of that likelihood function. That's the correct tree. Okay? So that brings us to our model. Okay? And this is kind of important, right? Because you will be examined on this, guys. So there's no holiday here. Okay? You're going to be examined on how to make trees. And so what you need to understand is that neither likelihood nor in the next lecture Bayesian, neither of them can work without a model. Okay? Neither of them can work without a model. So if you are busy writing in your exam, there is maximum likelihood, and then there's Bayesian, and you forget to say that they need a model. And you forget to say why models are necessary. And you forget to say what the model is. Then -5. -5. No. Don't drop marks, guys. Be thorough. Right? What is a model of substitution? Okay? Let's put it like this. So we know that about the observed data. Okay? We know that they're DNA sequences. We know about the trees. I've been telling you about trees now for a few lectures now. You know what DNA sequences are, and you know what trees are. But what is this model? You also know other models, right? I explained it to you. In population genetics, we use the Hardy-Weinberg model, the Wright-Fisher model, and the neutral model. You can call them theories. Hardy-Weinberg theory, Hardy-Weinberg equilibrium, Wright-Fisher equilibrium, neutral equilibrium, neutral theory. They're all models. Okay? So let's just stick to model. Right? So what is this model we use? we use this model to describe how a mutation happens. Okay? Because different mutations are what actually makes the DNA sequences different, right? You know that in some sequences there's an A and the others there's a G. So that means either an A evolved into a G, DNA polymerase made a mistake, added a G instead of an A, or the G evolved into an A, right? So it could have been either way. So that's what we need. We need a model that gives us the rules to tell us how the mutations happen, when the mutations happen, okay? How frequently do they happen, okay? And when we have this information, then we can work out how two DNA sequences, how closely related are they, and when they shared a common ancestor in the past. So we can't go anywhere without these rules. There's rules for nucleotide substitution. In other words, a model for nucleotide substitution. So consider a DNA alignment, okay? That means individual 1 is a line of DNA sequence. Individual 2 is a line underneath. Individual 3 is a line. So you have lots of A's, G's, C's, and T's, a sequence of DNA. And then another underneath it, and another underneath it. And they're lined up perfectly, okay? So that is called a DNA alignment, okay? And a DNA alignment, each position is a homologous position. And this is beautiful because why? You know from the early lectures that you can only use homologous characters to make a tree. A homologous trait is the only trait you can make. You can't use an analogous trait. So that's why each position on the alignment, we know it is homologous because it's descended from a common ancestor. We've lined up all these sequences and we're saying, well, that position has to be homologous. It's in the very same piece of DNA that all of these have in common. It's from a homologous piece of DNA and it's the very same position. It's a homologous position. So that's the beauty of using DNA sequences. Each position is a perfectly homologous character or homologous trait, okay? And you know now that means we can use any mutation in a DNA alignment to make a tree because they're all homologous positions, okay? So each substitution, that means each time there's a mutation, what happens to that homologous character or that homologous trait? It changes. It's the change, the state changes, right? So you can have the different states are what? A, G, C, and T, right? So all we need to do with the substitution model is describe the rate of the different characters change. How often do we go from A to G? How often do we go from G to A? How often do we go from C to T? How often, and so on and so on and so on. So that is how we work out. That is how we. That is what our model is all about. Okay, I'll give you some examples, right? So first example is that transitions and transversions do not mutate at the same rate. You know this already, okay? What is a transition? A transitional mutation is when a purine becomes a purine. It mutates, it changes, but it's changed to another purine, okay? Or when a pyrimidine mutates to another pyrimidine, okay? But transversion is what? Is when a purine becomes a pyrimidine, or when a pyrimidine becomes a purine. And those don't happen at the same rate. Transitions are much more common, whereas transversions are a lot rarer to find. So if you look at the genome, you will see many, many more transitions and many fewer transversions. So they don't occur at the same rate. If they occurred at the same rate, transversions and transitions will be equally frequent in the genome, but they're not. So how do we take it into, if we consider that they are equally frequent, we will give transversions the same weight as a transition, and then we will get our branch lengths wrong, and our tree will be wrong. Okay? So we need to say that transitions evolve at a different rate to transversions. And the way we do that is a simple way is to use Kimura's two-parameter substitution model, which takes this into account. It gives transversions a different rate to transitions. Okay, what about the fact that not all the sites in a sequence, in a piece of DNA, mutate at the same rate? Some sites may be constrained by selection. In other words, they evolve at a very slow rate. Some sites may not have selection. They may just be neutrally evolving, in which case they can evolve at a very fast rate. So this will affect what? The length of the branch, because the length of the branch, remember, is about the rate of evolution. So the length of the branch will change between any two taxa, the distance, yeah, the length of the branch will change, okay, depending on the rate of mutation. So we need to actually consider that there are different rates, okay? And so if you use a model that assumes the same rate, well, you will get the wrong tree. You will get branch lengths which are not correct. So you need to be able to make sure that the model that you are using takes into account the fact that different branches have different rates of evolution, okay? So, and that is called rate heterogeneity, and we usually model that using a gamma distribution. Okay, so that brings us now that you know what the observed data are, now that you know what the tree is, and now that you know what the model is, we can actually get into what maximum likelihood itself is. So, what is maximum likelihood? The principle behind maximum likelihood is that it evaluates a hypothesis, in other words, a tree. Each tree, and there are millions of different potential trees, each tree is a different hypothesis that we are testing using maximum likelihood. So it evaluates a tree about evolutionary history by calculating the probability that the proposed model and the hypothesized tree together will give rise to the observed data set. Okay? So basically, it's saying, it's calculating how likely is the observed data to have given me this tree, given my substitution model. Okay? We search for the tree that maximizes the likelihood of the data given a particular substitution model. So how do we calculate the likelihood? Okay? And here we go. First, we need to define a substitution model in the form of a probability matrix. A probability matrix basically gives the probabilities of changing from one base to another. In other words, it's the rules. I told you the model is the rules, and the probability matrix basically defines those rules. The rules of change from one base to another base, like from a G to a T, or from a C to an A, or so and so on. So the probability matrix can be written like this. M is equal to M, I, and J, where I and J are A, C, T, and G. Okay? So this probability here, Mij, is basically the probability of mutating from state I to state J. Okay? And I and J could be any unit. So it could be Mij could be the probability of mutating from a C to a T, or the probability of mutating from a G to an A. It could be a probability of mutating from any base to any other base. Okay? So that's the probability Mij. Okay? That's one mutation, one unit of mutation in one time unit. Okay, so now, knowing that, what are there are many different substitution models and different probability matrices. The more common ones I explained to you, I didn't actually tell you that the Jukes-Cantor is even more simple than the Kimura two-parameter model. The Jukes-Cantor model assumes that all bases change at the same rate. One parameter, equiprobable changes. In other words, one parameter, and you see it's alpha here, and basically this matrix is telling you the C will mutate to an A at probability alpha. A T will mutate to a C at probability alpha. A G will mutate to a C at probability alpha. In other words, the mutation rate for any base to any base, Mij, is alpha. It's the same. Okay? Whereas Kimura's two-parameter, as I told you, it takes transitions and transversions into account. So, where are the transitions? Here we go. From A to C, you are going from purine to purine, and therefore you have just an alpha. Okay? So whenever you're going from purine to purine or purine to pyrimidine to pyrimidine, like C to G, for example, you still have one rate. Okay? That's the transition rate. However, when you're going from purine to pyrimidine, yeah, or from pyrimidine to purine, then you have a different rate. And we multiply the alpha by another constant called kappa, okay, which then gives you the rate of transition of transversion. So from a G to an A, when you're going from a pyrimidine to a purine, it's the transversion rate. So it's two rates. You've got alpha and you've got alpha multiplied by kappa. So transitions, according to Kimura's two-parameter model, transitions are mutate according to alpha, and transversions mutate according to alpha multiplied by kappa. Okay, and then the most complex model is the GTR model. Okay, and this, look at this thing. You still have A, C, G, and T, but look, G, to mutate from a G to a C is the rate RGC. The rate from C to a G is RCG. And the rate from, basically, the rate from any base to any other base is unique. So it's not just saying transitions and transversions, it's saying every change has its own rate. Okay? So this model is very complex. This one is semi-complex, and this one is very simple. So if you made a tree using this model, you will make your tree very quickly. But if you make your tree using this model, it might take days, if not years. Okay? Because look how complex that model. Every change, every mutational change has its own rate. Okay? So, how do we calculate the maximum likelihood? Let's look at the tree below. Okay? There are 4 leaves. Okay? A, G, C, and T. Okay? So let's just have these, say that these are individuals with this sequence. This individual has, at the same position, okay, at one site, at a particular site, here are 4 individuals. This individual has an A, this individual has a G, this individual has a C, and this individual has a T. Okay? So, the gray circles, G, T, and T here, are the known ancestral bases. Normally, when we're calculating the maximum likelihood, we don't know these. We don't know these 3 here. We don't know them, okay? We only know this. This is what we've sequenced. We've sequenced 4 individuals, and at that position, they've all got a different base, all right? That's what we know. This is what we usually don't know. But for this example, let's pretend that we do know it, all right? So, what is the probability? In other words, what is the likelihood? What is the probability of observing our data? Here's our data, okay? Given this tree and our chosen model of substitution. That's what we're asking, okay? And how do we do that? We go, the likelihood of this tree is the probability of a T to a G, and how do we get this? How do we get this? We started the ancestor. The ancestor mutated from a T to a G. So that's the first likelihood, the mutation from a T to a G. Okay? Then what is the next one? The G goes to an A. Okay? And the G goes to a G. So here we have it. The mutation from a T to a G, the probability of mutating from a T to a G, that's this one here, multiplied by this one here, which is MGA, GA, there it is, multiplied by MGG, which is this one, the probability of changing from a G to a G. Okay? So actually, with these three terms, 1, 2, 3, we've worked out the likelihood for this part of the tree. Now let's look at this part of the tree here. Again, it's obvious, right? So how do we go? MTT, which is from the mutation from T to a T, and then mutation from a T to a C, that's this one here, M times MTC, and the mutation from a T to a T, that's this one here. You multiply that again. So the probability of the whole tree is all the probabilities of each and every step multiplied with each other. Okay? And that's all it is. That's how easy it is to work out the maximum likelihood. You basically work out the probability of change of every single step in the tree, and you multiply them by each other. And then the sum of all of that is the maximum likelihood. Okay, so now what happens when in the normal situation when we don't know the ancestral basis? Okay, here we go. Here it is. Same tree, but we don't know the basis. And we just put a placeholder X, Y, and Z because we don't know what the state was. Okay, in this position, what is the maximum likelihood? Also exactly the same, right? The likelihood of the data given this particular tree topology is the sum of X, Y, and Z, sum over X, Y, and Z, okay? And that is basically M X Y, which one is that? That's this one. M X from X to a Y, sorry, the M from X to a Y, multiplied by the M from Y to A, that's this one here, multiplied by the M, the probability of mutating from a Y to a G, there it is, and again for this half of the tree, MX to a Z, there it is, Z to C, there it is, Z to T, there it is. So it is exactly the same as our previous example, except we don't know the states. And instead of using the states, we use a placeholder names X, Y, and Z. Okay? So let us analyze a particular site J in our DNA sequence. A, this figure here that I'm circling with my mouse, that is supposed to be alignment there, sorry. This is the alignment here, okay? Here's individual 1, 2, 3, and 4, okay? And here's the sequence of individual 1, here's the sequence of individual 2, here's the sequence of individual 3, and here's the sequence of individual 4, right? But for site J, here's site J, at a particular site J, we have individual 1 and 2 having a C and a C, individual 3 has an A, and individual 4 has a G, okay? We're only interested in this site, okay? So for this tree topology, okay, for this rooted tree, we can say, okay, we'll put C and C. So for this topology, let's just assume that we are having a rooted tree, all right? But we can start with our unrooted tree, for example. We can start with our tree here where we have individuals 1, 2, 3, and 4. Okay, that's our unrooted tree. And in this situation, you would probably have individuals 1 and 2 most closely related, okay, because they both have the C, and then a distance separating them from 3 and 4, which are actually different, okay? But there are no ancestors in this unrooted tree. If we wanted an ancestral state, we would have to actually include these 2 extra positions on the tree. Remember, there's only 1, 2, 3, 4 here, okay? We will have to include this ancestor here at position 5, and we'd have to include this ancestor here of all of them at position 6, okay? That's the difference between an unrooted tree and a rooted tree. The rooted tree has the ancestral positions, whereas the unrooted tree, you don't know what is an ancestor and what is derived here, okay? Whereas here, you know exactly the ancestors are here and here. All right, so how do we work it out? So for this topology, we can work out, okay, the probability is the probability of going from a C, 2, and that's right. We don't know these two positions here, okay? We don't know these two positions. Let's go back one slide. 5 and 6, positions 5. We know these positions, but we don't know what the ancestors were. But we use maximum likelihood to reconstruct that, okay? So say the ancestor was an A here and an A there, okay? In other words, an A here and an A there, right? So then the probability of this would be what? Obvious, right? The probability of this would be M from C to A times M from C to A times M from A to A times M from A to A from M from A to G. Okay? You've worked that out based on the last example I gave you. But having two A's here is not the only possibility, right? But we've worked out the likelihood of this combination if there are two A's, right? Now let us assume that these are not two A's. Let's keep one of them as an A and make one of them as a C there. So let's calculate the probability of this tree, and that will be MC to C, MC to C, MC to A, MA to A, MA to G, all multiplied with each other, right? Then, but C and A are not the only possibilities for the ancestors, right? Let's actually, so basically you put every combination of different bases in these two positions of the ancestors, and you work out the likelihood of that particular tree. And then you put another combination of these two ancestors and you work out the likelihood of that tree. And you put another combination of ancestors and you work out the likelihood of that tree. And you add all of them up together. Remember, for inside each tree, you multiply this likelihood by that likelihood, but for each step you multiply, right? But when we're looking at the log with the probability for this tree topology, okay, we add them up, we sum them up, okay? So we sum up the log likelihoods for all the possibilities, okay? And that is just the likelihood for position J. Remember, it is just the likelihood for this position here, okay? Not this one, not this one. Each of them have their own. In fact, once we've done J, we have to go to this one. Then we have to go to this one. And every time we have to do this calculation, giving every single possibility. So you can see very quickly how maximum likelihood winds up becoming millions, if not billions, of different calculations. The more individual sequences you have. Okay? So as soon as all the possible trees have been evaluated, your analysis will find the tree that maximizes the likelihood. Okay? So you've calculated this here for every single tree topology combination, for every single ancestral combination, and for every single site on your DNA alignment. Eventually, you will end up with the maximum likelihood. However, if you have a small number of individuals, like how many did we have here? We had 4. 1, 2, 3, 4, plus 2 ancestors here. Okay, we had 4 individuals. So we had a rooted tree with 4 individuals, with 6 individuals actually, if you include the 2 ancestors. How many possibilities? There are 945 different possibilities. Just for this tree here. Just for this tree here at this site. There are 900, because we have 1, 2, 3, 4, and we're rooting the tree 5, 6. We have 6 positions we need to calculate. Okay? So here, S, we have 6 positions. There are 945 different possibilities. Each one has a likelihood. We have to find out which of the 945 has the highest likelihood. That's not a problem. There are only 945. Okay? Because there's 6. However, what if I said to you, okay, all the individuals in our class, okay? We are about 15 in our class. Let's make a maximum likelihood tree for all our individuals in our class. Look how many possibilities. Let's go to 10. 15 is somewhere between 10 and 20, right? Let's assume 20, right? There are suddenly 8.2 times 10 to the 21. Do you know how big that figure? It's like that, taking that decimal there and moving it that way 21 positions. That's more than trillions, okay, of different possible trees. So now, finding the maximum likelihood among trillions of trees is not so easy. When you only had 945, it was easy. But now you have trillions. It's not so easy anymore. Okay? And then what happens when you say, okay, 100 individuals, look at how many possibilities there are. And maximum likelihood has to do the calculation for every single one. Okay? What do you think is going to happen if we try to use maximum likelihood with 100 different individuals? Not 6 like our last example, but 100. And 100 is not many. It's not even one population. If we want to make a tree with 100, it's not even a big complicated tree. And yet, to do it through maximum likelihood, it will have to do this many calculations. 10 to the 184. So you move that decimal 184 places. 3, 4, 0, and 184 zeros after it. That is a massive number. I don't even think that there's actually a way to say that number, except like this. So in other words, maximum likelihood will take forever to find that tree. It will probably never find it. It will calculate for years, if not lifetimes. It's not possible to calculate the maximum likelihood for so many combinations. Okay? So the advantages then of maximum likelihood before we end the lecture. The advantage of maximum likelihood is that it has a lower variance, much lower variance than other methods. It's statistically the best method to use. Okay? They are robust over many violations in the substitution model. So even if your model is not 100% correct, it doesn't matter. Maximum likelihood will find the same tree. There's no long branch attraction like there is in neighbor joining. So likelihood almost always finds the best tree, even with a short piece of DNA sequence, right? Maximum likelihood will do much better than parsimony and definitely better than distance. It will find the right tree. Okay. However, you already know that there are not going to be there are not going to be a few disadvantages when we get to disadvantages. Okay, so the likelihood method is the best statistical method we can use. It's the most thorough. It calculates every possibility, and when you calculate every possibility, the probability of every possibility, then the one with the highest, that's the right tree. Okay. The problem is that it is too many. If you have too many sequences, then to calculate the maximum likelihood is enormous. Okay, so the disadvantage of that is that maximum likelihood is extremely slow, and it takes many big CPUs to calculate maximum likelihood. Okay, you have to have a very, very fast computer, and this, what I'm going to come to now, it often needs a heuristic search to explore tree space efficiently. Now, what on earth does Prof mean by that? Well, that is what we call the Markov chain Monte Carlo search method in tree space. We'll get to that not in the next lecture, but in the lecture afterwards. But I just want to say that this heuristic search means that it's not possible to actually calculate the maximum likelihood of every single possibility, because you saw how many possibilities we would have to create. Even if we had 100 sequences, it is impossible to physically calculate the maximum likelihood. You will be doing your PhD for 100 years. Okay, so in order to get around that problem, we use what we call a heuristic search, which is a Markov chain Monte Carlo, and that way we can still use maximum likelihood to find the best tree, even if you have more than 100, okay, individuals, because it doesn't have to calculate each and every probability. We will get to that heuristic in the next 2 lectures. Okay, and of course the result can be dependent on the evolutionary model you select. So you also have to take some time to choose the best substitution model. Okay, it's not, you can't just say, I'm just going to use a Kimura 2-parameter, or I'm just going to use a Jukes-Cantor. You have to first test the data to see which model is most appropriate and use the most appropriate model. Okay, so those are the advantages and disadvantages of maximum likelihood. And remember, as I keep saying to you, how trees are constructed is going to be examined. Okay, you need to know this. You need to know the methods. You need to know the principle of each method. You need to know the advantages and disadvantages of each method compared to the other methods. You need to be able to tell me all the ways of making trees. Okay, and next lecture, lecture 28, we will be looking at an even better method than maximum likelihood to make trees. Okay, and that will be the Bayesian method, and we'll get into that in the next lecture.